Welcome to you all. Thank you for turning out on what is a lovely evening to join us in the last of this year's Executive Dean's lectures. For those that don't know me, I'm Alistair Burt, Executive Dean of the faculty. I'd like to start by acknowledging the Dana people, the original custodians of the Adelaide Plains and the land on which the university's campuses here in North Terrace, Waite and Roseworthy are built. So thank you, as I say, for coming along. And we're going to hear from two speakers this evening, Dr. Luke Selth and Dr. Theresa Hickey. Luke's research is focused on understanding the mechanisms underlying prostate cancer progression and metastasis, spread of prostate cancer. And he has a specific interest in the roles of what's called the androgen receptor and microRNAs in these processes. He was previously a recipient of the South Australian Young Tall Poppy Science Award and heads up the prostate cancer research program within the Dame Roma Mitchell Cancer Research Laboratories. Theresa seeks to make discoveries that can rapidly enter clinical testing by investigating drugs that are already approved for use in people, so it wouldn't have to go through all the approvals processes, which can be time-consuming, but yet these drugs are used for other purposes and not currently used to treat breast cancer, but could quite effectively. She was a recipient of the Royal Adelaide Hospital Research Foundation Career Development Fellowship, a highly competitive award. So Luke and Theresa will discuss how they leverage the similarities of breast cancer and prostate cancer, particularly around hormone receptors, to make discoveries aimed at revolutionising the treatment of both diseases, attempting to reprogram the hormone receptors that drive both of these malignancies. So to discuss transforming endocrine therapy for breast and prostate cancer, I'm pleased to welcome Luke and Theresa.
I think I'm going to talk here and grab this. I think.
Well, you can, that's fine.
Can everyone hear me okay? Please. So the reason I'm not on the lapel is that my colleague Luke and I decided that we're going to do something a little different and do a tag team talk. And we're doing that in part to highlight the similarities between breast and prostate cancer and to highlight how we work together and learn from each other to really make discoveries that are relevant for both diseases and can be mutually beneficial. So we hope it works and we hope you can follow along. And I am going to start off with some statistics showing you that even statistically, these two diseases are very similar. Prostate cancer is the most commonly diagnosed cancers among men, as breast cancers are the most commonly diagnosed cancers among women. 1 in 7 men will develop prostate cancer by their 85th birthday, and 1 in 8 women will do the same by their 85th birthday. In terms of mortality, they're very similar as well. It's estimated that over almost 3,500 men will die of prostate cancer by the end of this year, and likewise, it's estimated about 3,200 women will die of breast cancer. So over 6,000 people in Australia will die of one of these diseases by the end of the year. Now, our lab, the Dame Roma Mitchell Cancer Research Labs, which is run by Professor Wayne Tilley in the audience here, has been really unique around the world in looking and investigating these two diseases in parallel. And indeed, Professor Tilley inaugurated the first PAC-RIM breast and prostate cancer meeting that took place here in Victor Harbor in 2003. This was an international meeting series that continues to this day and is extremely popular. We get requests all the time for wanting to join into this meeting. Professor Tilley was also the initiator and senior author of this review in Nature Reviews Cancer that came out in 2010 that really, for the first time, put forward the perspective that breast and prostate cancer are more similar than different in the terms of their pathology. So I'm going to start with the breast. Breast is best. And just give you some primer on normal breast biology. The breast is a system of ducts that end in glandular tissue that can produce milk at lactation. And this development begins around the age of 10 years old and pretty much reaches its adult form by the age of around 15. Now, this pubertal breast development is exquisitely dependent on the action of estrogen hormones in the female. And so estrogen is required to drive invasion of this duct from the nipple back towards the chest wall. And estrogen is also critical in these glands developing off of the ducts and then maturing to structures that can produce milk if needs be. Now, estrogen acts through a very specific protein called the estrogen receptor, as do all sex hormones and sex hormone receptors, in which the hormone binds to the receptor to activate it. And there are 3 sex hormones and corresponding receptors in the body. And these are the estrogen receptor, the progesterone receptor, and the androgen receptor that mediate the actions of estrogen, progesterone, and androgen, respectively. These receptors and their hormones have a lot in common, and they function in very similar ways, whereby hormone binding activates the receptors, and once activated, the receptors bind to DNA at very specific sequences called hormone response elements. And when this occurs, it occurs upstream of target genes to regulate their expression. And these genes will send out messages to the cell to tell them what to do, to grow, multiply, to die, or to make milk proteins, for instance. Now, while estrogen receptor and estrogen action is important to drive normal breast growth and function, abnormal estrogen receptor action is what drives the large majority of breast cancers. Indeed, at least 75% of all breast cancers are driven by the estrogen receptor. And here's just some images showing you what a normal breast structure looks like compared to a breast cancer. In the normal breast, you see these structures that are actually ducts, open ducts, and over here you see those structures are absent. Now, these images have been processed in special ways, whereas the estrogen receptors appear as green dots. There are very few single green dots in the normal breast, even though it's exquisitely driven by estrogen action. However, over here, you see in the breast cancer, these green dots, these estrogen receptors, are everywhere expressed in those abnormal breast cancer cells. Now, because the estrogen receptor is what drives breast cancer, the main therapeutic strategy is an anti-estrogenic one. Historically, that meant removing organs in the body that produced estrogen, such as the ovaries and the adrenal glands. Later, drugs were developed that could block the action of estrogen receptor in the breast cancer cells, and more recently, drugs have been developed that prevent your body from producing estrogen.
Okay, let's switch to the prostate for a minute, shall we? So, like Theresa did for the breast, I want to put this in a bit of context by telling you a little bit about the normal prostate first. So for those who don't know, the prostate is this small gland here surrounding the urethra underneath the bladder. And it actually plays an important role in male fertility. So it's a secretory gland that's important for male ejaculation, and indeed about 30% of ejaculate is comprised of the prostatic fluid, which feeds and protects the sperm. Now, like in the breast, androgens, and the most well-known of these being testosterone and its more active metabolite dihydrotestosterone, and the androgen receptor are absolutely required for the growth and development of this organ. So if you don't have DHT or you have an issue with your androgen receptor, you will not develop a prostate. And again, like the situation with the estrogen receptor in the breast, what the androgen receptor actually does upon binding to its ligand, in this case testosterone, is it associates with these response elements on DNA and it regulates the expression of a set of target genes. And in the normal cell, these target genes give instructions to cells to differentiate, but also to produce secreted proteins. And probably the best known of these is PSA, or prostate-specific antigen. So, I've just told you that the AR acts as a good guy in the normal prostate. Again, analogous to the breast, what happens in prostate cancer is that AR switches to a bad player. So what happens is that it no longer regulates these genes that are involved in differentiation and that can actually inhibit growth, but it starts regulating a different set of target genes. And these genes can actually now promote growth, promote survival of the tumour, and promote metastasis in some cases. Metastasis being the spread of these prostate cancer cells out of the prostate. Because of this exquisite dependency of androgens and the androgen receptor for the growth of prostate cancer, the mainstay therapies for advanced prostate cancer are all targeted towards the androgen receptor. And I'm going to tell you a bit more about these on the next slide. So basically they're comprised of two main classes. The first are inhibitors of androgen production. So these are your classic hormone therapies that inhibit androgen production by the testes, but also by the adrenal glands. And it also includes newer generation androgen biosynthesis inhibitors like abiraterone, which is really focused on inhibiting production of androgens in the adrenal glands, but also within the tumour. And the end result of these therapies is lower levels of circulating testosterone in the blood. Now, the other main class of therapies to treat advanced prostate cancer that target the AR are AR antagonists. And the way these drugs act is to directly block binding of testosterone to the androgen receptor. And they actually outcompete androgens for binding to the receptor. So the end result of these two classes of drugs is a non-active androgen receptor. So either there is not enough androgen to activate it, or these drugs can actually block the binding of androgen to the receptor. And so the result of this is that it doesn't target these androgen response elements, and its target genes are switched off. Now I want to tell you a little bit about how resistance to these therapies develop. The unfortunate reality in prostate cancer is that therapies that inhibit the androgen receptor are never curative. They always fail, often after it can be a considerable period of time, but as I said, they're never curative. And so to explain how they fail, I need to show you a picture of the androgen receptor that shows you a little bit more detail of the protein. So this is a protein, this is the androgen receptor, and it's comprised of these three main segments. The NTD, which is the N-terminal domain, the DVD, which is the DNA binding domain. This is the part of the protein that allows the androgen receptor to interact with DNA. And the LBD, the ligand binding domain. This is the part of the protein that actually binds androgen. And as I said, all of the therapies that we currently use are targeted towards inhibiting this part of the AR activity. So the inhibitors of androgen production and the AR antagonists. Now, some seminal work done by Professor Tilley, who's in the audience, who's the director of our larger laboratory in the mid-1990s, as well as other groups around the world, identified ways that the AR can get around these therapies. So you have to remember that cancer cells are clever. They come up with ways to evolve and adapt to the therapies that we treat them with. And one classic therapy resistance mechanism that we see in lethal prostate cancer is the emergence of AR mutants. So these little red lines are to designate mutations of the AR in the ligand binding domain. Now what these mutations do is twofold. They can either convert these AR antagonists to agonists. So these drugs that we're using to block the activity of the AR actually now start to activate the activity of the AR. The other function of these mutations is to enable the androgen receptor to be activated by other ligands, so non-androgenic ligands. So now it can be switched on even in the absence of androgens, as you might see if a man's being treated with androgen deprivation therapy. More recently, probably less than 10 years ago in fact, our group and others identified another flavour of androgen receptor that emerges in lethal prostate cancer. And these are what we refer to as AR variants. So you'll see immediately these are very different. These completely lack the ligand binding domain. So the therapies that are used to target this part of the protein are no longer effective. These, in essence, act as constitutively active forms of the androgen receptor. So by that I mean they're active all the time, even in the absence of androgens. And they're now thought to be a key driver of this lethal phenotype.
So a similar scenario is true for breast cancer, and I won't repeat what Luke had just gone through, but basically we know that those cancers that are ER positive and have become resistant to all the different forms of anti-estrogenic therapy available are those that kill the majority of women with breast cancer. Now, we've lagged behind the prostate cancer field a little bit in the sense that ER mutations and splice variants, similar to those that Luke just talked about for the androgen receptor and prostate cancer, were detected also in the 1990s, but they were largely ignored because they were very rarely found in primary cases of disease. If we take it now 20 years onward, and with the development of technologies where we can sequence the DNA to great depth within tumors, and we've looked again at not only primary tumors but metastatic tumors, now we see that in these tumors that develop despite repeated treatment with anti-estrogen hormone therapies, that quite a large number of them now have acquired either mutations or variations of the estrogen receptor. And just like Luke was saying, the ones that have been found so far, these mutations focus on that domain of the estrogen receptor that binds hormones. And currently, the function of these mutations in terms of altering ER action is a current field of study. We still don't know what some of these mutations actually do. So in that respect, our knowledge of mutations of the androgen receptor in prostate cancer is advanced. The thing is that along with the ability to sequence tumors and detect these mutations, some really major developments in the breast cancer field were taking place in terms of modeling. So I'm going to talk a little bit about how we model breast cancer. Now, breast cancer, as well as a lot of cancers, including prostate cancer and many, one of the kind of main tools that have been used are cell line models. And these are cells that were originally derived from a patient sample, and they were trained to grow within culture flasks as a 2D monolayer. And they basically grow as weeds because you give them nutrients to allow them to grow. And these cell line models are very useful tools, especially to test ideas. But cells growing in the bottom of a plate don't mimic the complexity of a primary tumor. And so you can get interesting information from them, but that information will not directly lead you to discoveries that you're going to test in the clinic. And so the modeling, I'm going to jump over a whole lot of other things, but this is the current sort of gold standard that's been developed in the field of breast cancer, and these are called patient-derived xenografts, or PDXs. And this is where tissues from either the primary tumor or metastatic deposits are removed from the patients, put into mice, and developed to grow and expand. And some of them can be serial passaged in mice, sustain a certain stability, and can be used as models. They're very hard to establish. Sometimes it's taken years to establish certain models, and they're very expensive in terms of time and money. However, this kind of modeling is likely to lead you to discoveries that are much more clinically relevant. And as an example of that, it was using these PDX models of endocrine-resistant breast cancer which really revealed the role that ER mutations play in driving endocrine-resistant disease. Now, while people were working on PDX models, we were working on another kind of model in the Degrading labs. And this model appears more simplistic, but is very powerful, in which we take pieces of tumor directly from surgery, from the clinic, chop them into little pieces, and layer them on the top of a gelatin sponge. And this sponge is then submerged in growth media to which we can add treatments. And this feeding of the tumor from below sustains the tissue architecture and the complexity of the tumor piece. We've shown that it sustains the pathology, the original pathology of the tumor, and it sustains a functional hormone signaling axis. So we developed this as a very good, different alternative preclinical model that we've now have used in several published studies and that we've shared with people around the world now. And the advantages are that we can interrogate short-term treatment responses right within a patient's primary tissue. This saves time and money. These experiments are much cheaper. And it also reduces the use of animals. And this fact captured the interest of the press recently. I think it was 2 weeks ago. A story went around the country where this word sponge tumors first arose. So this was the press kind of characterizing our model. Now, what we're doing currently is trying to expand the usefulness of this model. We first developed it using breast tissues. Then we moved to show that prostate cancer tissues also grow well as sponge tumors. And what we're doing currently is that we're trying to put the whole lot together in which we can also take tumors that have been expanded in mice and cut them up and put them into our sponge tumor system. And in that way, we can test a whole lot more treatments in a more rapid, cost-effective way than if we were to do that same experiment in mice. It would take hundreds of mice and a lot more dollars. Now, we developed the sponge tumors together with collaborations with our local oncologists and surgeons. And as I said, we've shared this with groups around the world. And in turn, we work closely with groups around the world who develop the PDX tumors, and we have a very nice collaboration going in that.
Okay, so now I want to tell you a little bit about prostate cancer models specifically. As in many of these situations in breast and prostate cancer research, prostate has been lagging behind somewhat. But as in the breast, what has become apparent in the last couple of decades is that cell lines are really not cutting it in terms of producing information that's clinically translatable. And so with that in mind, we have been collaborating with a fantastic group at Monash University in Melbourne on developing new prostate cancer patient-derived xenograft models, so these PDXs that Theresa described. So they have a really world-leading program where they're obtaining tissues from warm autopsies or from biopsies and putting these straight into mice and basically trying to grow these as new PDXs. And you can see the scope of this program here on the slide. So they've enrolled 69 patients in this program. From those patients, they've obtained 138 tissues. Many of these have been exposed to androgen deprivation therapy, that's ADT, or ADT plus some of these newer AR targeting agents, so they're very clinically relevant models. And what we've been able to show, we've been sort of doing some of the more mechanistic analysis of these models, which has been really, really illuminating. Because they have established a warm autopsy program where they're obtaining multiple metastases from a single patient, we've been able to look at those metastases and to find differences between metastases that have come from the same patient. And this has actually led to some very exciting findings. The key one being that the mechanisms of AR-driven resistance in terms of AR mutants and AR variant expression can be different in different metastases from the same patient. They've also enabled us to identify new resistance mechanisms, and of course, very importantly, the key use of these new models is to evaluate new therapeutic targets. And with that in mind, I want to now take you through a couple of brief vignettes on new therapeutic targets that we have been investigating in our lab in prostate cancer. Before I do that, I just need to take you back to this slide and have a look at this structure of the androgen receptor again. So you'll remember that all the therapies to date target this part of the protein. But you'll notice that this N-terminal domain and the DNA binding domain are parts of the protein that are conserved between all of these different forms of the androgen receptor that are emerging in lethal prostate cancer. And so our idea was to try and target this other part of the protein, this N-terminal domain, which to date has been very tricky to drug, but we have some exciting new findings that I'll tell you about in a minute. Before I do that, I just need to tell you a little bit about its function. So in terms of a very simplistic view of it, this part of the protein is responsible for turning on and off the AR target genes and is therefore indispensable for its activity. In a bit more detail, what it does is it serves as an interaction surface for other proteins called AR co-regulators that are absolutely required for AR activity. So if AR can't interact with these, it cannot function. And to cut a very long story short, our group, in collaboration with colleagues in Texas, University of Texas Southwestern in Dallas, have been able to develop a new lead compound called a peptidomimetic that can actually block the interaction between some of these co-regulators and the AR N-terminal domain. And we propose that it will be active against all of these forms of the AR that emerge in lethal prostate cancer. The name of this drug at this stage is M25. So we've been testing this drug in some of these new PDX models that I just described. In one of these models, obtained from a brain metastasis and grown subsequently in mice as a PDX, we were able to culture this in the unique ex vivo explant system that Theresa described. And using this system, we're able to show that this new drug can inhibit the growth of these tumours. So you don't need to know what this factor is, KI67. All you need to know is that is a marker of growth. And so you can see that when we treat these pieces of tumour that are derived from this patient metastasis with the drug, we get reduced growth. Similarly, this is a marker of cell death. And you can see when we treat the tumour pieces with the drug, we're getting increased death of the tumour. So it's looking very promising at this stage. It's looking even more promising when we consider that this is the current standard of care agent, and this is actually a drug that was used to treat this patient. And you can see that this drug does not suppress the growth of the tumours. This drug is enzalutamide, which is an AR antagonist, and it does not cause death of these tumours. So it's perhaps not a surprise that it was not able to control the growth and progression of this man's lethal prostate cancer. The second vignette that I just want to briefly talk about, again, is a lesson that we've learned from breast cancer. So in breast cancer, these drugs called selective estrogen receptor degraders, so these are not inhibitors, these actually degrade the AR. The most commonly used being fulvestrant, have been widely used to treat breast cancer since the early 2000s. But there are currently no degraders of the AR in clinical use, and given their efficacy in breast cancer and this huge similarity that we've been talking about between AR and ER in the prostate and breast, we considered, are we really missing a trick here? So we've been collaborating with a company called GTX, who are based in the United States, and they've developed a new selective AR degrader. And this SARD actually is targeted towards, again, this N-terminal domain of the protein. So we think it will be effective against all of these forms of the AR that emerged in lethal prostate cancer. And we actually have some very nice data that's just been published in Cancer Research a few weeks ago supporting this hypothesis. So this diagram is what we call a Western blot. You don't need to really know too much about that, except that this band is a measure of the androgen receptor. And you can see that when we use increasing amounts of this SARD, we're getting degradation of the AR. This, in fact, is an AR mutant. So this is a cell line that's driven by this AR mutant. This is a different cell line that's driven by the normal AR, but also an AR variant. So this is one of those flavours of AR that lacks the ligand binding domain. And you can see again that the drug very effectively degrades these proteins. So it's showing a lot of promise in these cell lines. Of course, I've just told you that cell lines are not ideal. So how does it work in vivo? This is one of our patient-derived xenografts. You can see that the SARD inhibits the growth of this PDX, and it also inhibits the growth of this more conventional xenograft as well. So it's a very exciting finding, and as I said, just published, and we're hoping that it will move forward in the clinical spectrum shortly.
So similar kind of work is happening, looking for new drugs to target estrogen receptor directly in breast cancer. But I want to now introduce another new concept and where we have been putting some focus. Although new drugs that target estrogen receptor may be effective, they fall within what I call the hammer approach. Basically, all of them are aiming to wipe out estrogen receptor and its action within the body. Now, we've been doing this for over 100 years. It was Beatson who over 100 years ago took the ovaries out of women and showed that their tumors would stop growing. And so this kind of anti-estrogenic approach has been around for a long time. And the problem with this approach is that it seems like no matter how we go about knocking down ER, it keeps popping back up again in endocrine-resistant disease. That is what's lethal to the patient. So more women die of endocrine-resistant ER-positive breast cancer than any other form of cancer. So while we develop new drugs to hit it harder and harder, it seems to just keep popping up. The other thing that's often not talked about is that female bodies don't like to lack estrogen receptor action. Indeed, most women that take these anti-estrogenic drugs feel quite miserable. There are a lot of side effects, including hot flushes, severe joint pain, fatigue, depression, endometrial problems that might require a hysterectomy. And in many cases, it's now known that a lot of women stop taking these drugs, often quietly, because they don't want to let on that they're feeling so miserable. So not only is treatment resistance an issue with anti-estrogen therapy, but also quality of life. And we're looking to not only improve survival, but make life bearable for those that continue to have disease. Now, so to do this, we're thinking of a new treatment paradigm, that instead of wiping out and annihilating estrogen receptor action, can we rehabilitate it? Can we push it around towards acting in a better way? And the general approach we're to doing this is by activating other sex hormone pathways that we know will interact with estrogen signaling in breast tissues. And one of the reasons we're focusing on this is that sex hormone receptors, there's a lot of drugs that have been developed and approved to modulate the action of sex hormone receptors. And so this comes to what Professor Burt was mentioning, is that if we can find a new use for these drugs to treat breast cancer, we can translate our findings very quickly. And here's an example of that. Our first evidence for this ability to rehabilitate ER came from this paper where we showed that we could activate progesterone receptors to reprogram ER activity in breast cancers. This is a paper that came out in 2015. It got a lot of press, and it really established the proof of principle that we can do this, that we can take a bad ER and make it better, make it behave better. Importantly, because there are a lot of drugs around that can stimulate the progesterone receptor, we very rapidly went to establishing 3 clinical trials within 2 years of publishing this study, which was quite exciting. And this is just a diagrammatic representation of what we found. So like Luke was saying, for the androgen receptor in prostate cancer, in breast cancer, the estrogen receptor binds to regions in the DNA that are associated with poor outcome genes. These are the genes that are telling the cancer cells to grow and multiply and thrive. However, what we showed is that if the tumor has progesterone receptor and you treat with progesterone, is that activated progesterone receptor interacts with the estrogen receptor over here and can drag it to new places on the DNA. And what we found was that these new genes were genes that were associated with better outcomes, that were associated with slowing down or stopping growth or stimulating cell death. Now, the caveat of progesterone receptor therapy is that not all ER-positive tumors have progesterone receptor. Indeed, in that paper, we reported that about 22% of all estrogen receptor-positive breast cancers do not have the progesterone receptor. The good news is that the androgen receptor is there in almost all of them. Here is an example of 2 PDX models that we've been working with. This brown dots here are cells that are positive for the factor up here. So these brown dots are all estrogen receptors, these are progesterone receptors, and these are androgen receptors. And this is just an example of 2 PDX models. Here's one that have all 3 receptors. Here's one that just has the estrogen receptor and the androgen receptor. Now, for time, I can't show you all the unpublished data that we have showing that androgen receptor can also reprogram estrogen receptor to produce better outcomes for breast cancer. But what I will show you is just this bit of data. It's an example of a PDX model developed from a metastatic carcinoma in a young woman. This one had all 3 receptors, the estrogen receptor, the progesterone receptor, and the androgen receptor. You can see this model was stimulated to grow by the addition of estrogen, which is the line in blue. On the top, I show you the experiment where we treated with progesterone, which is in red, showing you that treatment with progesterone opposed the action of estrogen in promoting growth of this tumor. The second graph down here shows you the same, where we activated androgen receptor with dihydrotestosterone in this instance, and also very potently inhibited the ability of estrogen to stimulate the growth of this model. So just to summarize, that what we're trying to do here is forge a new treatment paradigm where we're not trying to annihilate estrogen receptor action, we're just trying to modify it and change it from being the bad guy back to being the good guy. So changing it from a poor prognosis and reprogramming it either with progesterone receptor activation or androgen receptor activation in order to produce better outcomes. Now, I've mentioned the 3 trials we have underway with progesterone receptor activation. We have also initiated 2 trials with a new drug that very selectively activates the androgen receptor in breast cancer cells, but does not cause virilizing side effects. You would never treat a woman with dihydrotestosterone unless they want a gender transformation.
So what I hope is becoming apparent is that we're trying to learn from the other disease type to inform our research. And a very obvious question then, given these exciting findings that Theresa has just presented on reprogramming the estrogen receptor in breast cancer, is can we do this for the androgen receptor in prostate cancer? Now, I've told you that in the normal state, AR is a good guy, so it promotes differentiation. It actually blocks growth of the normal prostate. But in cancer cells, it switches to a bad player, which I described earlier. So again, learning from the breast, can we reprogram AR back to this more normal-like state? And would this yield a more durable therapeutic response? Importantly, like in the breast, would this be associated with fewer side effects? So a big issue, as in breast for anti-estrogen therapies in the prostate, is anti-androgen receptor therapies are associated with really severe side effects. So impotence, fatigue, depression, to name just a few. And if we could avoid or alleviate some of these side effects with a better targeted AR therapy, men would be much more likely to want to take those therapies. The problem is, in the prostate, is that we're lagging a little bit behind in terms of understanding how this reprogramming might work. So we don't actually know what other hormone receptors or what other factors we could activate to reprogram AR. So with this in mind, we have recently established what I think is a very exciting project to try and identify factors that can reprogram AR by doing a high throughput screen of thousands of different hormone receptor ligands and other drugs to try and identify factors that can, as Theresa described, push AR from the bad outcome sites to the good outcome sites. So this work, very excitingly, was just funded by both the National Breast Cancer Foundation and the Movember Foundation in their inaugural breast and prostate cancer linkage grant. And so the focus of this grant, it's not just on prostate cancer, it's obviously on breast cancer as well, is really to get away from this hammer approach that is initially effective but associated with poor quality of life, but even more worse, associated with resistance that drives the tumour to a lethal state, and try and reprogram the ER and AR back to a more normal-like state that may be associated with better survival outcomes, but also, very importantly, improved quality of life. Now, these types of major research programs don't happen in isolation, so while this work is being led out of Adelaide by Wayne, Tilly, Theresa and myself, we work very closely with the group I mentioned at Monash University before, who developed these fantastic new prostate cancer models. We also work closely with a group at the Garvan Institute of Medical Research. And also in this particular project, we have collaborators and advisors, an outstanding team from around the world. And I really think this team will enable us to achieve fantastic outcomes in this grant.
So just to sum up, this is the current paradigm with our current approach to targeting AR and ER using anti-hormone therapies, endocrine therapies for breast and prostate cancer, because the disease trajectories are very similar for both diseases, that when you hit this state, we can get disease progression. Given one form of the drug, it progresses another form of the drug until you get very aggressive disease that kills the patient. And what we're trying to do is to cut off that trajectory right here. In essence, by not depriving the body of androgen and estrogen action, but modifying it, and hopefully under that scenario we'll be able to truncate that trajectory and increase survival, with a caveat that we also hope to reduce the side effects that the anti-estrogen and anti-androgen therapies incur. So this is our new paradigm. And with that, I want to thank the many people in our lab. We have a great team. Here are some of them, not all of them. Our collaborators from around the world, as Luke mentioned before. We've been fortunate to receive continual funding for this project, and we hope that continues into the future. And last of all, I want to say a special thanks to patients who have donated tissues to do this research. But also, we have a number of advocates, patient advocates, and other community members that we liaise with in order to make sure that our research is communicated to the general public, but also so that the general public and interested parties can comment and input into our research directions. And in particular, the focus on reducing side effects was very much driven by that communication.
Okay, just before we get into the Q&A, a couple of final plugs. So, for those of you who aren't aware, this is the 1st of November, the month formerly known as November. Who here knows about the November Foundation? Hopefully everyone. It's a fantastic charity that's focused on saving and improving men's lives through projects focused on prostate cancer, testicular cancer, and also more recently mental health and suicide prevention. We have a team. Our team is called the Movember and NBCF linkage, Mo Bros and Mo Sisters, a bit of a mouthful. If you want to donate to Movember, donate to us or donate to someone else, please do so. And finally, to celebrate our recent launch of the new grant that we were just describing at the end of the talk, we're having a Pink Mo breakfast 2 weeks from today. It's in this building on the 3rd level in the staff lounge. It's going to be from 10:30 a.m. till 12 p.m. and anyone is welcome to join us.
With a raffle and bake-off.
With a raffle and bake-off as well. Thank you very much.
Thank you both very much indeed. This is open for questions. Maybe I could start with a question you might not expect. So you've been focusing understandably on breast and prostate cancer and those hormone receptors, but these hormone receptors are expressed in other tissues. Are there messages here in terms of tumour growth in other common cancers?
Absolutely.
Absolutely. In fact, we put in a grant for that to Bupa a couple of years ago, and we weren't quite successful in that one yet. But yes, absolutely. In fact, steroid receptors affect almost every organ in the body. There are very few that don't. Sex hormone action could, for example, explain the differences between liver cancer in men and women, for instance, and it definitely has a broad
Yes.
Yeah.
Certainly from an AR perspective, in recent years there's been a lot of interest in AR's role in other epithelial cancers, such as bladder cancer and others. So we are certainly thinking about how these types of strategies could be applied to other cancers and hopefully improve outcomes in other diseases as well.
Many, many years ago, we published a paper on androgen receptors in liver cancers, liver tissue and liver cancers. It was a pretty probably uncited paper, but yeah, that's why I was kind of aware of it. All right, yeah.
I don't know if I need the mic, do I? Thanks, guys. That was really terrific. I really liked how you went back and forth between the two topics. I want to draw you both out, perhaps, but I guess especially Luke on when you go back to the origin of the cancer and you talked about the good guy turning to the bad player. Are you talking about androgen receptor switching to find different response elements, and what happens to cause that?
Yes, so we're talking about different sites on DNA that it's binding to and then activating a different gene expression program. There's a whole suite of things going on. I mean, the initial mutations that caused the cancer to develop in the first place obviously changed the molecular dynamics in the cell quite a lot. And so there'll be changes to co-regulators, there'll be changes to epigenetics in the cell, and a whole lot of other factors that are impacting where AR binds. Certainly in terms of co-regulators, we think they're very important. So we do know during the development and progression of prostate cancer in particular, you get gain of particular co-activators, so gain in their expression and their activity, and then also loss of key co-repressors. And that certainly does change AR's DNA binding profile. So yes, that's what we're talking about, changes to the DNA binding profile, and there's a lot of different factors that control that. and Theresa might want to add some
Yeah.
more from the breast.
So the same as in the breast, so I didn't have a chance to put it in, but we know now that, for example, some breast tumors are very, what we call indolent. They've probably grown within the breast over 30 years. They're not likely to kill a patient, but because we can detect them, with mammography we can take them out. We now have data showing that the way estrogen receptor binds in those tumors is very different to the more aggressive forms of tumor. And in fact, what we showed in that reprogramming paper is that in those aggressive tumors where we treated with progesterone, we're changing that binding profile from an aggressive profile back to that more benign profile. Yeah, and like Luke says, that's likely to be involved with co-regulators and a whole host of other mechanisms.
Great. Yes. Question back there. The microphone's coming.
That was really great, guys. I had two questions, one for each of you. Theresa, so androgen receptors is quite highly, expressed in a lot of those breast cancers. So treating with the progesterone to change the action of the estrogen, how does that actually influence the androgen receptor? And is it good or bad?
Yeah, we don't know that. That is the topic of a grant that we got funded for last year, and that's exactly what we're doing right now. So we have a whole lot of data on activation of the progesterone receptor alone and estrogen receptor alone, and that current focus of a lot of our work is to look at them together. Sorry, I'll end there before I go to.
And I was just curious, your M25 drug and your SARDS, have you tried them in combination to see if it has an additive effect?
Very good question. I mean, there is a lot of interest, obviously, in combination therapies. Obviously, a lot of the therapies that we're talking about in therapy failure, it's single agent therapies, and that's why the idea that perhaps using combinations would be more effective. In that particular scenario, we have not tried those two in combination. We've tried both of them in combination with current standard care therapies. And we do that because often, in terms of the clinical pipeline, you need to compare these with and against, the current standard of care. I think it's a great idea. They both work in very different ways, so that's probably something that we should try and do.
Question, yeah.
One of the troubles for prostate cancer screening is PSA, as you said, with breast cancer, no one knows what tumours are going to develop into something nasty and what's going to stay indolent for 20, 30 years. So, and it's been all sorts of work with different PSA types and all that sort of stuff, as you know. But could the change be something to do with androgen receptor modification, or could it be something else, or could it be a mixture of those things which determines whether something's become nasty or not?
Yeah, that's a really good point. I mean, today the talk was pretty much all at the other end of the disease spectrum, sort of advanced, very aggressive forms of prostate cancer. We also have a research program in our group that's really focused on trying to differentiate those indolent versus aggressive primary tumours. And as you say, at the moment, that is just a real problem. And so that causes, obviously, a lot of over-treatment, but also under-treatment of the aggressive cancers. So PSA is not the answer. I think we all know that now. Whether we can use AR as a readout of identifying the indolent and aggressive tumours is a great idea. This is certainly something we've spoken about. And so one idea we've had is to try and develop an assay that can look for this movement of AR to different sites on the chromatin. So the sites are associated with more aggressive and poor outcomes. And so, an assay like that might be able to tell us if a particular tumour has aggressive AR signalling versus more normal benign AR signalling. We have not been able to go too far down that path. This is obviously quite new work, but I think it's a fantastic idea that certainly we're exploring.
One last question.
Okay, Theresa, is there any possibility that
So in the triple-negative breast cancer, so for those who don't know, triple-negative breast cancer is the most aggressive form of breast cancer. And one of the reasons it's aggressive and it causes a high rate of death within that small group is that it lacks all the targets. It lacks the estrogen receptor, it lacks the progesterone receptor, and it lacks the HER2 receptor. And so none of the current targeted therapies are effective for this case. So androgen receptor, about 20 to perhaps 30% of triple-negative cancers have the androgen receptor. And androgen receptor in that space has been marked as a potential bad guy. And there is some evidence, mostly with cell lines and particularly with one cell line, that suggests AR is a bad guy. And I should say the AR in that cell line is a mutated AR. However, we have several projects going on now with other models of triple-negative breast cancer that have AR that show that in some of those tumor, AR is still a good guy. And so we are developing studies now. We do studies in that space trying to determine whether we can tell whether AR is a good guy or a bad guy in triple-negative breast cancers and develop an assay that will say, should we be inhibiting AR or stimulating AR in that scenario? So that's a great question. Thanks.
So I think we'll need to wrap up there. I'd like to, I agree with Darryl, Luke and Theresa, you made a great tag team, a very engaging presentation, and you've demonstrated that these two tumours are more alike than different. I think it's also reinforced that highly effective science benefits from having researchers who might have slightly different starting points in terms of their interest, but the synergies between them can lead to mutual benefit and to great outcomes. So you've already demonstrated all the collaborations you have internally but also externally, and the reason I'm kind of rushing here to the end is that they're just about to get a taxi to the airport to go and meet their collaborators interstate. So thank you both for that wonderful presentation. Thank you all for coming. That is the last Executive Dean's lecture of the year. There will be, of course, more in next calendar year. We hope to see you again at that. I'd hate to say a happy Christmas and New Year at this stage. It's just a bit too early. But thank you all for your attendance. Safe home. Thank you.